Okay, the first thing we have to do is label our test tubes 1 and 2. To each tube, we add 5 milliliters of water. To tube number 1, we're going to add 10 drops of water using a dropper. To tube number 2, we're going to add 10 drops of the sugar solution. To each tube, we're going to add the testing reagent. For sugar solution, it is the Benedict solution, and we're going to add 20 drops. Add the same thing to tube 2. And we notice the color. So we can see the color for both of these is a light blue. Now, Benedict solution works in warm temperature. So we're going to take these 2 tubes and put them in a water bath. Okay, while that's heating up in the water bath, we can complete the rest of our experiment. Here we already have our tubes filled with our 5 milliliters of water. But to tube 1, we still have to add our 10 drops of water. And to tube number 2, we're going to add 10 drops of our potato starch solution. You might want to give this one a shake before you add it. Make sure everything's dissolved. 20 drops, okay, so we not have to add another 10 drops to this one. We want to keep it consistent. So that was a total of 20 drops, and now we're going to add the total of 20 drops of our starch solution. Then we're going to add some iodine. Iodine tests for starch. So we're going to put in 4 drops of our potassium iodine. And let's make a quick note of the colors. So we'll gently swirl these around. And we see, okay, we can see the color. We see a color change in around 30 seconds. We'll be able to see the color change. You can already see it start to happen. Tube number 1 we see is yellow amber. Tube number 2 with the starch, we can see it's already beginning to turn brown. That's what happens when you mix potassium iodine with starch, it turns brown. Next experiment, our lipid test. So what are we going to do? We've already got our 5 mils of water in our tubes. Now we add 20 drops of water. Why do we add 20 drops of water? 20 drops, 20 drops. We remember we want the same volume. Now we're going to add 20 drops of our oil. Now we have to add our detecting agent, our Sudan 4. We're going to put in 20 drops. 19 and 20. Oh, that one, 22. We'll put in 22 in here. 1, 2, 18, 19, 20, and to be consistent, 21 and 22. And then we gently give these a shake. Okay, here you can see the red color goes throughout. We'll give a shake with this. And what do we notice? The red color stays in the top. We'll see what that means in a little bit. Now we go to our next test that tests for our protein. Protein test is completed with our Biarritz reagent. So again, we put in 10 drops of water, drops of our albumin powder. Albumin is the powder, or is the protein that we find in eggs, the egg whites. Ten drops in both tubes. Now we put in our Biarritz agent, which looks for 20 drops. We'll notice the color. It's a pretty blue, like my blue eyes that you can't see because of my glasses. Now tube number 2. We'll give it a quick little stir and see what color this changes into. So now we have to figure out our unknowns. How do we do it? We perform the exact same test we did with our knowns. So we can label our test tubes 1, 2, 3, and 4. And we just put in, let's see, what does it say? Our unknowns. Let's put in 10 drops of our unknown into each test tube. Give it a quick shake. What do we do next? Just complete the test. So number 1, we did our sugar test. Okay, what do we need to add? We add our, let me just double check, 20 drops of Benedict solution to tube number 1. After our 20 drops, we put it in the water bath. Then we do our second test, our iodine, which was here. And then we put in 4 drops of our iodine. 1, 2, 3, 4. Now we complete our next test using our Sudan 4. Sudan 4, we need 20 drops. Just like we did with our other solution, we give it a quick little shake. And last but not least, we do our, where'd it go? Test using our protein test using the Biarritz agent, and that we add 20 drops. Now what do? We repeat the exact same test. And again, how do we know it worked? Well, now we're looking for color changes. So here are the unknowns. Now you figure out unknown number 1 is which of our 4 tests. Unknown number 2 is which one of our tests came back positive.